Welcome back, viewers. So, having established in the previous video the crucial importance of genes, of the genetics, because that codes for protein, and protein is basically everything, muscle, pigment, etc. In this video, I want to talk you through what you might call the hierarchy or structure. So what I mean by that is it's how things fit within things, right? So in geography, you'll understand that we have the world, and that's divided into continents, and we can divide those into countries, and countries have regions and towns and so on. And so we're going to look at, I think I mentioned it last time, of the genome, which is kind of like the recipe book. And in fact, let's stick with this analogy because I think it might help. So we have imagine you have a restaurant, right? And everything you make in that restaurant, the soups and the puddings and everything, is in a single recipe book. And I've said before, that's kind of like the genome, all the instructions to make all the proteins to make a starfish or a sunflower or a Labrador. So we have the recipe book of the genome, all right? If you were to look at a cell under a microscope, this is a cell, a eukaryotic cell, we'll come on to that another day. You'll see the cell and you'll see a nucleus. And if you look at the right sort of time, you'll see in the nucleus these funny little things. And if you put a stain on your cell, these things will become colored. And these things are called chromosomes, which literally means the chrome bit refers to color. Chromosome, like monochrome means sort of one color, but I suppose it's black and white. Chromosome means colored body. And that's a little bit like the recipe itself, right? So we've got a recipe book. No, it's not. It's like the chapter, let's say, because you might have a chapter on soups. You might have a chapter on, I don't know, fish or who knows what. So our recipe book is kind of like the genome, and our chapter is kind of like the chromosome, which is that thing there. Maybe I'll color code this. So that's the genome is kind of like everything, all of that thing, and then the individual chromosomes are those things. Okay. Then the next level down, I suppose, in the chapter would be the recipe for French onion soup or mulligatawny or whatever it is. So then we've got the recipe. We might have that on a page, perhaps, which that is equivalent to the instruction to the gene, which is the instruction, like we saw last time, the recipe for making, the melanin pigment, or for making lactase, the enzyme for breaking down the sugar in milk. All right. If we were to look in a bit more detail to these chromosomes, you'd find actually that it's not a little X. It's actually made up of lots and lots and lots of this polymer, DNA, right? So I'm going to sort of do it like this. So a chromosome is to the DNA polymer what a jumper, a sweater, is to the yarn, to the wool, right? So, you can talk about it as just as yarn, but when you knit it together, you make the jumper. And so this is the chromosome, but it's made up of tightly wound DNA, that double helix we saw before. So if I were to look at a particular stretch, let's say, that's not a good color, is it? If I were to do like that and sort of blow it up, we would see this sort of double helix, and that's our thread. Okay, so hopefully that makes sense. And indeed, we could pretend that that stretch is the stretch for whatever we want, growth hormone, let's say. So let's pretend that is the stretch for growth hormone, and there's our gene. Okay, so hopefully this is clear. We've got the recipe book, which is the genome itself. We've got the individual chapters, chromosomes. This isn't really true because in in a recipe book, you'd have, like I said, all the soups in one chapter and all the, um, you know, sweet stuff in another chapter. In the chromosomes, it's not really like that. You don't have all the pigments on one and all the enzymes on another and the hormones on another. It's all interspersed. So that doesn't really work, but it helps perhaps understand. Then I suppose a recipe is made up of individual words, and those words are made up of letters. All right. In this case, our gene is made up of the thing I've drawn, the double helix, is what's called a polymer. So the monomer is called a nucleotide. I'm afraid let's see, nucleotide. I'm afraid there's a lot of jargon in this, but I don't think they're too scary, these ones. Deoxyribonucleic acid, this is a nucleotide. And those nucleotides it's not like in English where we have 26 letters. We've got different bases with a nucleotide, and I'll explain this now. So if we were to look at that, so nucleotide, I might go onto another page actually. A nucleotide, the monomer, a single unit, we have a lot of this in biology, we have monomers and polymers like amino acids and proteins, is made up of 3 different bits. So we have a thing called phosphate, which is made up of the element phosphorus and oxygen. Plants need phosphorus, that's quite important, NPK fertilizers. So you'll understand, therefore, plants need phosphorus, phosphate, in order to grow because they need that for their DNA. And that links to another thing which makes the job a bit difficult because different syllabuses, different specifications call it different things. So sometimes it's just called a sugar, but properly it's called deoxyribose. That ending, of course, helps us out. Sugar, O's ending, like glucose, lactose, cellulose, etc. It's sometimes called pentose because you can see it's a pentagon. It's got 5 corners. O's again for the sugar ending. It's a little bit like glucose. Glucose is more of a hexagon. It's got 6 carbons. But anyway, each corner, we just represent this as this is a schematic. We just draw it like this. We don't draw all the detail. And then the other bit would be the base. And that's the interesting bit because that's the bit that can vary. Okay, so that is 1 unit. There would be another one above. I'll just draw in gray. And obviously one below because it's a chain of them. Okay. And in fact, there'd be one coming along the other way because it's a double helix. So I could draw one here, like so. It's a bit tricky to do upside down. And it's not the same. But you get the idea, and it would be going on like that. Okay. So this particular bit is the nucleotide, a single unit. So it's the monomer. That's the general word, the monomer, and it's called a nucleotide. Phosphate, and I've not labeled this, have I? The base. But that's the bit that's interchangeable. So you may already know this. I think it's in general knowledge that we've got A, C, T, and G. And you can also see from the way I've drawn it that the grayed out corresponding nucleotide that it's connected to has a bit of a lock and key arrangement going on there, what's called complementary. So you have, let's draw it like this. So we'll draw that one, and I'm going to call that C. And then I'm going to draw this one, and I'm going to call that G. So C and G fit together. And we also have another one, which is, let's call that one A, and then let's call this one T. Okay, so they're called cytosine, guanine, adenine, and thymine. C links to G, and A links to T. And this is called, so these are the bases. So my nucleotide has a C base, but they can have G or A or T, but the one it's connected to has to therefore be G. And they fit together, and it's called complementary base pairing. This might be a bit more information than you need in certain specifications, but maybe better to have too much than too little. And you can see that comple bit, like the word complete, because they fit, one completes the other. It's the E spelling, because if you have another word in English, complementary, like you're looking nice today, but that's with an I, that type of complementary. Okay, so hopefully that's clear. Um, what else have we to say on this? So we've got the hierarchy. So we've got the genome, chromosomes, the gene. We could have other genes. So, any stretch on here, that could be the gene for eye color, for example. Or that could be the gene for an enzyme of some kind. Okay, like protease, amylase, something like that. So that's where everything sits. But one individual part would be a nucleotide. So, it will be just sort of that part. It's kind of like a ladder, and the sides of the ladder are made up of the phosphate and the sugar. And then the rungs of the ladder, the bits going across, are made of the two bases. I'm sure you'll find better pictures than mine, but hopefully it gets the idea across. And an implication of this is quite straightforward, that when when it's time to replicate, your double helix, let's draw it maybe just like this. 1, 2. So that's A, that's T, that's C, that's G. You can unzip. So you've got one strand, and then the other strands will come in. Whoops, that will go with T, A. So you can make another strand because of this complementary base pairing thing. So you take the double, unzip it, and then other bases will come in, and an enzyme will sort of join them together. So you go from, I suppose I should have drawn it like that. So let's say we've got this. That's our starting point. And then we have this complementary base pairing thing, and then we end up with this. You see, so the black would be the original, and then we've created a new pink, like so. So this is how you have DNA replication. And I'm going to leave it there, and next time we'll talk about how you actually get from the gene to the protein, as we saw.